Okay, thank you, Marty. So let me, while he switched the screen, let me tell you a little bit about two facts.
He's putting it right back.
One of the facts, I'm sure you know, the second one may not. But I'm sure you know that when you look at your body, your skin, which is the largest organ, that's everything that is inside. You know that, right? Now, inside your body, the largest organ is the vascular endothelium, and it's all over inside your body. And vascular endothelium is nothing else than your capillary system that is full of endothelial cells. That's why it's called the vascular endothelium. Is the largest organ inside your body, and it protects that all your organs work properly. So what I'm going to talk about today is what happened with this when the vascular endothelium gets injured in your lungs or in your kidneys, or even the vascular endothelium in your skin or in your brain. What happens is you develop a fibrotic disease that could be lethal. So APIE-Therapeutics, what we are working is in the next generation of anti-fibrotic drug therapeutics. So let me share a little bit about us. If I could get, okay, here we go. Okay, so a little bit about APIC. We are located in the Research Triangle Park in North Carolina. So I, do all the way from NC to here to meet all of you face to face, and hopefully some of the virtual attendants, I will meet them. And so at some point, so what we are focusing is in diseases where the vascular endothelium gets injured and become dysfunctional, and fibrogenesis is part of the process of developing fibrosis. So the target that we are going after is the apelin receptor activation. This receptor, what it does in your body is repair and regenerate the vascular endothelium and also stop the fibrosis processes. This was discovered and well patented all over the world in the Research Triangle Institute, which some of you may be familiar with it, and they are the discoverers of many of the commercial drugs that are available. Our first, I wish I can tell you, we have a list of all the diseases we want to go after, and certainly we can't. And we have to focus on one disease. And we are focusing right now on one that you probably know it as scleroderma, but the name is systemic sclerosis, interstitial lung disease. The reason we are selecting this disease, I will get into it, but as of today, we already have been granted by the FDA an orphan drug designation for this disease, and this disease have the ability that once we put the proof of concept in the lung fibrosis, we can expand this therapy to other diseases. So there's an opportunity to create a value, and I actually like very much this morning presentation by Min when she talked about getting and investing early, and this is where we are. So let me tell you a little bit of scleroderma. And I have friends that have a scleroderma. You probably heard too when this comedian not too long ago passed away, and he was very involved in the scleroderma world. So what happened is this disease is a chronic disease, by the way. It involves a build-up of collagen because your vascular endothelium gets damaged, and your body goes to, I have to fix it. And it goes into this excessive build-up of collagen that becomes fibrotic tissues. The issue and the challenge with this particular disease is that it could be one organ, but it could be multiple organs. I'm sorry?
Nothing.
Okay. This disease is classified as limited or diffuse, limited meaning one organ, diffuse meaning you have involved multiple organs. And the key organs are your microvasculature, your kidney, your lungs, and your heart. Lungs, which is our main focus area, is the most involved organ, and you have about, when the moment they tell you to the moment it becomes lethal for you, it's about 2 to 5 years. And this is true for pulmonary fibrosis also. The treatments for systemic sclerosis are basically treating your symptoms, and there's no disease modifying drug for systemic sclerosis. So it's an extreme medical need as we speak. So let me just show you how usually when a patient start feeling something and they go to the doctor, usually what happens is they start losing the blood in the extremes. And that's called Raynaud syndrome. You probably have heard about that. From there, progresses very fast. Your vasculature capillaries start, getting dysfunctional, and you start, it looks like RA, but it's not RA. And you start losing a lot of the flexibility that you have in your skin. This is a manifestation of a skin fibrosis, and you could last about 10 years if you don't develop lung fibrosis. But once it progresses to your lung, it becomes really, you have very little lifespan. And I know you cannot see the lungs of this, beautiful lady here, but this is a picture here that you can see to your right, how a fibrotic lung, looks like, and if you can think, you cannot breathe. Your lung is not exchanging the air with the rest of your body. So a little bit about scleroderma, then I'm going to jump to, what we have, why we believe, what we have is novel, and it will translate to human. A little bit about the disease. This, it affects all races. Usually, if you're going to have the disease, it will show between your 30 and your 50s. There's a 4 to 1 woman impacted, and 1 of 3 of these patients die, within 10 years. And, juveniles, which I was actually very sad to find out, you could develop the disease, before your 20s. And there's a prevalence of about 100,000 and 2.5 million in the world. And this is what allows us to qualify for orphan drug designation, which is a really an awesome way to work with the agency for a fast approval. And there's an economic burden, on these for the patients and the family. So let me tell you what we are doing. So I told you already, when you have an injury in your vascular endothelium, your body responds, right, by creating inflammation and immune responses that then it tells the body, okay, there's something wrong going here. Let's go into a production of collagen. And this system is called a maladaptive healing system because it's trying to heal, but it goes into a chronic maladaptive process that now you are developing, these fibrotic and scarring your tissues. The cartoon that you see there, if you start from the left, just imagine that's your lung. It's a healthy lung, healthy capillaries surrounding, your organ. You could get a virus, a bacteria, actually COVID-19, unfortunately. It is a good example because you've heard a lot of people developing lung fibrosis or kidney fibrosis. But it could be caused by other things. It could be your own immune system attacking you. Once you have that dysfunction in your vascular endothelium, your body goes to the next step, which is produce this maladaptive process. If you look at what the medical industry, the medical healthcare and the pharmaceutical, they have been going after fibrosis, for a couple of decades, right? The idea there has been, let's stop the collagen build up, let's stop the inflammation, let's stop the immune response. However, all of that is only in the downstream of the disease. So you probably will agree that part of why we have not find success and patients, even with some of these treatment, continue progressing if that is not really stopping the disease. It's not modifying the disease. So what APIE-Therapeutics, and this is really what got me really excited to get involved here, is the activation of this receptor will work at the upstream, repairing and regenerating the vascular endothelium, but it also will work at the downstream, stopping the fibrogenesis, which is part of the collagen production. And so it is a novel way, and I'll show you some of the data. So I'm going to start talking about APT-101. We have a portfolio of about 800 drugs, and we are, narrowing, focusing on 3 lead candidates, and our priority candidate is 103, and it is positioned to address a disease-modifying effect on systemic sclerosis, ILD. And then this is the kicker here. Because I told you already a few minutes ago that these patients develop fibrosis not only in the lungs, they're going to come to the clinical trial already with a skin fibrosis, and they will come with already vasculopathy, which is the vascular endothelium dysfunction. Some of them will come also with kidney fibrosis. So what this does, it would allow us in the trial to look at the treatment, not only for the lungs, but also systematically to these other fibrotic endpoints. And all of that it does is just add more value to the asset and just set the asset to launch into expansion of other therapeutic opportunities. So I'm going to show you 2 data points. These data points, this is a model to look at the vascular endothelial permeation. This is a human cell model, was done in the University of North Carolina, UNC, in Chapel Hill. And I think the picture talks for itself, right? On the left-hand side, these are 2 devices or 2 models where the vascular endothelium was stimulated with pro-inflammatories, which we know will start the dysfunction and they start leaking, right? The next 2 pictures that you see to the left is 2 different doses where you can see the APT-101 was able to repair and regenerate the dysfunction and bring back, total integrity to that vascular endothelium. This is what we believe will stop the fibrosis progression. The second point that I will show, the second data point that I will show you is this is actually testing for collagen buildup, right? So we know we can target repair and regeneration of the vascular endothelium, but what happened in the downstream of the disease? And this is what we saw. I'm sorry. Now I don't know how to go back. Sorry, apologies for this.
No worries.
Yeah, we are in family. We are in family office.
No worries.
Okay, good. Okay, so data point here. If you look to the left, you have a healthy group. This was a study done in mice. It is good to have collagen, so don't get me wrong. Having collagen is a good thing. I want to have collagen in my face, and when it gets down, I try to put a little bit to keep it up. So it is a good thing. However, when you go into a chronic injury, which is what this model do to provoke fibrosis, now your body goes into this overproduction of fibrosis. If you look to the right hand of the plot, what you see there is perferon. That is one of the medicines that get given to people that develop lung fibrosis. It does reduces the collagen buildup, but APT-101, which are the 3 middle bars, reduces particularly at a dose of 100 and 125 significantly. It reduces and inhibits by 77% of the collagen buildup. That's awesome. And we were very excited to have that. Now, if you look at the tissue, because you could look at this data, but now looking at the tissue by a vet doing histology, these are three tissues look but for different things. The top one is showing you with a biomarker of fibrosis, untreated versus treated. And you can tell, the improvement in fibrosis by, the color brown not being as obvious in the group that was promoted with the disease. The second one is another way of measuring collagen through biomarkers. And same thing, we saw an inhibition of the collagen build up. And the third one, which was actually a really good measurement to do, is looking at, cell death. And that's part of also a pathophysiology in the fibrosis. So it prevents the cell death. Talking about competition, I see what it is. This has a delay.
Yes.
I thought.
Yes.
Okay. So when you look at the competition, and what I wanted to highlight here, if you look at the mechanism of actions that are being pursued, are actually very similar. And all of these mechanisms of actions are in the downstream of the disease. So it is not addressing the upstream of the disease. And some of the trials have stopped, have not shown, progress. So what APT-101 is doing, the competition landscape, it really has a competitive edge by bringing to the clinical trial a novel mechanism of action. And it will be the very first disease-modifying drug treated in clinical trials. And coupled to that we expect that we will see more than just lung fibrosis reduction, but we will see also reduction in the other fibrotic organs in these patients. So moving to how we are thinking and what we are, how we're going to, the fundings that we are going to raise and how we're going to the milestones to increase the value. Here, the first thing I wanted to show you is, we raised $9 million. Some of that was non-diluted from the NIH. And we have done, all the steps that, from patent this globally to scaling up to commercial manufacturing, all the way to send a pre-IND package to the FDA. And we received a favorable review and comments from the FDA. So we know that what we have developed as a program, has a high success or a high potential success to be approved to go to clinical trials. The first $6 million of a $35 million will get us to finish conducting all the IND GLP tox, which is the most expensive studies, and we will also repeat the manufacturing process to have clinical trial material. The next, in that series, the next 25, what it will take us is to conduct the phase 1 and phase 2 clinical trials to have the multiple endpoints. And what we are driving to is to have an exit to our phase 2 trial and have a partnership with Big Pharma that then can take it to phase 3 and commercialization. The funding that we have raised came from the North Carolina Biotech Center and the Research Triangle Institute. The IPF Foundation was an interesting one because when I called them, I was not asking for money. I just wanted to give them hope that people are looking to bring new treatments to their patients. And next thing I know, they called us and they wanted to participate in some of our notes. We have a Danish investment firm that also got excited by the difference in this mechanism of action. I have invested myself, board members, and then we have a couple of individual investors. And I wanted to share with you a quote of Jonathan. Jonathan, he's a practitioner of lung fibrosis, but he's also a researcher at Vanderbilt. And all he does is looking for how lung fibrosis happens. And when he started talking to us, the first thing he said, Esther, this is different than all the antifibrotic therapies. And I'm like, no, I know, but if you tell me that you believe in this, that's more validating. And I quoted right here from what he wrote to us, because this mechanism of action is attacking the disease before it starts. It's attacking from the upstream. So we have some, we have the validation that we were looking for. This slide is your typical slide of how we're going to, use the funds. I will not spend much time here unless you have questions.
We got about 5 more minutes.
Okay. Yeah, I'm wrapping up. So the way we are going to create value, this little picture here, what I'm trying to show you is, we're working with the Research Triangle Institute. They're doing the core discovery. APIE is doing the actual clinical development for this drug with the idea of creating value for the pharmaceutical company. And, there are several examples, and I particularly shows examples of preclinical at the stage that we are, and some early phase 1. So there is definitely opportunities to get the pharma industry excited about this. And, we have been talking to, a lot of the pharma, the 4 ones that I called that I'm very interested on having an exit with them. But, so those for the rest, and we are creating these relationships and getting validation of, our MOA. This is the team, and I think what I can tell you, I was the senior VP of global manufacturing at Biogen, and I have a career of 28 years doing just this, drug development, and I have the pleasure of, and the blessing of seeing over 13 drugs that have made it to commercialization and changed patients' lives. And you have a question or telling me?
I have a question. Do you get exclusive rights to the research coming out of Research Triangle?
Yes. Yeah. So we have.
Can you talk about the size of that research, but also can you talk about the funding that you've received so far, if any of it is non-dilutive, things like that?
So we have, out of the 9 million, 3.5 is non-diluted. Actually, I'm sorry, let me take that back. 7 million is non-diluted. 3.5 came from NIH, the other came from Research Triangle Institute. 2 million is diluted.
Is this a biological or a small?
It's a molecule. It's a small molecule.
And do you have a patent issued on it?
So we have patent on composition of matter, method of use, and other. And we have the patents issued in the 6 big market, tier 1 markets. And we selected 5 tier 2 markets that we also have put protection around.
So you elected to go for scleroderma in lung fibrosis.
Mm-hmm.
Why not go for interstitial fibrosis of the lung and primary disorders?
Yeah, so that's a good question, and we have been asked that. The benefit that we see with this patient population is that we can get multiple shots and multiple endpoints out of one trial, right? Because if we go just with patients with ILD, that's all we're going to be able to prove. Here, we already know these patients will come with skin fibrosis, will come with vasculopathy, and will come, some of them, not all, will come with some sort of kidney. So if you can imagine, we want to prepare a package that pharma's going to say, wow, I can take this drug. I could continue in scleroderma, but I can also, go to phase 3 and expand this into lung fibrosis, ILD, even kidney, if they wish to. So this is just here our board of director, Gene Greenwood. I don't know if you guys probably know him. He's the probably most known person. He used to be the CEO, a congressman, and then a CEO of the organization Bio. And, and we have, I don't have time to go over, their pedigrees, but, we have an awesome scientific group and clinical group behind us. So I'm going to wrap up, and hopefully what you can appreciate from everything that I said is this primary disease that we are going to go after, I mean, it's a very extreme unmet need, and the lifespan is not much. And in APIE, we have positioned this asset to have a successful IND, and with the translational medicine that we have done, we are projecting a successful human.
Just before you wrap up, did you talk about use of proceeds of this? I had the upper side. She did? Okay, great. Well, fantastic then.
Okay. Thank you. If you don't find me, you can find me. I will find me.